Wow. Okay, thank you very much for the introduction, and thank you very much for the invitation to come and speak as well. Yes, so I did a postdoc in the group of Kylie Vincent in the University of Oxford, and I'd like to talk today a little bit how I'm taking some of that work that we did in Oxford, and we're taking it in slightly different directions, or sort of taking things a little bit further, maybe. And this broadly can be described as this, so the right title here, where we're taking an electrochemical approach to allowing us to do a little bit of spectroscopy, which then will probe some structure and dynamics for metalloproteins. So if I get my introduction, my acknowledgements out of the way first, in case I forget later, most of the work that I'm going to show today was done in and around Leicester by a PhD student, Rufus Frew, but heavily in collaboration with Patricia, Zoe and Wang Jie, and also Stephen Carr from the Vincent Labs in Oxford. And just to prove that I actually do occasionally go to these places that I always list, there we go, that's a beautiful morning of me looking at diamond light source just at the beginning of an experiment, and then sort of some slightly crazed photos of me at the end of a week there, not looking quite so good. But okay, I'm going to talk a little bit about how we're combining electrochemistry and spectroscopy to probe structure and dynamics, ultimately, of metalloproteins. But I would like to caveat this with a massive, this is very much a work in progress talk. So I fully acknowledge that we might not quite get all the way to some of the results. Principally, I say metalloenzymes, but I'm principally interested in nickel ion hydrogenases. We're all familiar with these, reversible production or oxidation of dihydrogen at this tuned buried active site, with electrons getting in and out somehow via this chain of iron sulfur clusters. The active site is over here on the right, so we have a nickel and an iron joined by two bridging cysteine sulfur ligands, two terminal cysteine sulfurs to nickel, and then three biologically unusual, or biologically unusual unless you're talking to anybody other than this audience, I guess, carbon monoxide and cyanide ligands. And my screen has frozen. I don't know if it's frozen for everybody else as well.
Yes. Yes, as well.
Oh, excellent.
I had the same problem. You have to start from scratch. I think it's better.
Okay, I will do that then. Okay. All right. Apologies whilst I kill PowerPoint.
Okay. Yeah. Just try again.
Yeah. Apologies about that. Where am I? CCC. Okay. So this is the joys of the fact that I'm working at home this afternoon, because the heating broke in the lab this morning. So it was a choice between good internet or being warm.
Seems to be reversed from what we heard before. Everybody else is in university because there's the heating on.
Yeah. Okay, hopefully we're back now.
Yes.
Good. Right. Apologies about that. So, like I was saying, I'm principally interested in studying nickel ion hydrogenase enzymes, and we're going to start off with the classic sort of infrared part of the talk, making use of these carbon monoxide and cyanide ligands, and it really doesn't like this presentation at all. Okay. I'm going to try again, because I presume that this is frozen completely for everybody else as well.
So it's not working, it's not changing the slides.
Yeah. Okay.
Maybe you can start with the slide just after this one.
Believe me, I would love to. Come on. Okay, there we go. Right.
If you don't have any fancy animations, you could also quickly render a PDF and use this one. Smaller typically.
True. Okay. Hopefully this is working again then. So, yeah, so I'm going to start off with a little bit of a story about infrared spectroscopy using these carbon monoxide and cyanide ligands to try to tell us something about kinetics and mechanism, if you like, in the nickel ion hydrogenase catalytic cycle, particularly to do with sort of proton coupled electron transfer, which we shall see shortly. So in the interest of trying not to break this, I'm not going to turn my laser pointer on, but on the left of the screen here, there's just to orient those of us that might not be so familiar with FTIR spectra of nickel ion hydrogenase active sites. There's just an exemplar in the oxidized inactive, what we call the nickel B state, on the left of the slide here, where we have an essentially single CO band, because this is an essentially pure state, and so that's important because it means we have one state equals one CO band. So if we see multiple CO bands, we must have multiple states, and vice versa. Everything on the left is the cyanides, which I'm going to ignore completely, essentially during this talk. Okay, so beyond the active site, there are a number of conserved, in some cases highly conserved, amino acid residues, and I've pointed out some of the closest and most probably mechanistically relevant ones here. So we've got this glutamate 28, and this is all relative to E. coli Hyd1, which is very, very close to one of the terminal cysteine thiolate ligands to nickel, and this is thought to essentially gate much of the proton transfer activity in and out of the active site. I might disagree as to whether it gates all of it, but I'm not going to go into that today. Then there is a pendant arginine with a guanidinium head group positioned more or less directly above what we'd like to think of as this sort of vacant site in between nickel and iron. It's approximately the length of a hydrogen molecule away from the bridging position in the active site, and so there's some discussion of whether that plays a structural orientational role or as part of some sort of frustrated Lewis pair type mechanism during catalysis. So other differences beyond the active site, so you've got hydrogen oxidation, which takes hydrogen, gives you 2 protons and 2 electrons. So the protons have kind of got somewhere to go through all this amino acid architecture. How about the electrons? Well, the electrons transferred from the active site or to and from the active site via a relay of iron sulfur clusters, and the iron sulfur cluster closest, so the first electron exit point from the active site, if you want to think of it like that, it's called the proximal cluster. And if we loosely talk about nickel ion hydrogenases in 2 groups, for the purposes of this talk at least. So we have some oxygen tolerant membrane bound hydrogenases, and we have some oxygen sensitive membrane bound hydrogenases, where from a structural viewpoint a big difference is in the structure of that proximal cluster, but not only the structure, the number of redox states that are stable within that proximal cluster. So in the case of the oxygen tolerant membrane bound hydrogenases on the left hand side of this slide, we have a reduced oxidized couple, which essentially have very, very similar structures, and then we have an oxidized to superoxidized couple, where the transition to the superoxidized state involves the opening up of the cluster, a big structural change, and so it's a much slower process. In contrast, the oxygen sensitive membrane bound hydrogenases on the right hand side just have reduced and oxidized states that are again structurally pretty similar. A point that I'll come back to later is the fact that the redox potentials of the proximal cluster in the oxygen tolerant and the oxygen sensitive cases are really rather different. In the oxygen tolerant membrane bound hydrogenases, the redox potentials for all of the transitions from reduced to oxidized to superoxidized are close to or above 0 volts, whereas in the case of the oxygen sensitive membrane bound hydrogenases, the potential of that reduced oxidized cluster, for the sake of a hand waving argument, is in the ballpark of the hydrogen couple at pH 7, let's say. In terms of a mechanism, a catalytic cycle, we have the oxidized inactive nickel B state that I showed you an infrared spectrum of earlier. If we reductively activate that using either electrons or hydrogen, we can get into the catalytic cycle. So if we think in terms of hydrogen oxidation, I always think in terms of hydrogen oxidation just because the particular hydrogenase, hydrogenase 1 from E. coli that I study, is strongly biased towards hydrogen oxidation. So apologies if this is a little weird way to think about it for particularly the iodine hydrogenase guys out there. So if you think of the this nickel Si state as our most oxidized catalytically active resting state, so the sort of the greenish state in the middle there, that interacts with hydrogen. Hydrogen is split heterolytically to a proton, which goes somewhere, and a hydride, which remains bound in that bridging position between the nickel and the iron. Then the proton from that heterolytic cleavage of hydrogen is lost along with an electron to the proximal cluster to generate the nickel C state at the bottom from nickel R. And then there are essentially two pathways that we can take from there. We can either take a directly concerted proton and electron transfer to regenerate nickel S directly from nickel C, or, as is pretty much the thinking these days in most cases, we can take either a sequential or a concerted step where we go from nickel C to a nickel L state, where in most people's drawing of the nickel L state, this proton that I've put floating around the place would be maybe sat on one of these cysteine sulfurs, and then you've got either stepwise or concerted loss of that proton and the second electron from hydrogen oxidation to regenerate the nickel Si starting state. A small comment to make here is that there are multiple versions of the nickel R state and there are multiple versions of the nickel L state, and as yet, at least to my reading of the literature, there's no real consensus about how all of these various states are involved in catalysis, and in part that's because I think a lot of the experimental work, a lot of the kinetic experimental work that's been done on nickel iron hydrogenases in particular tends to focus on one particular system, and so you only ever really see the catalytic characteristics of that one particular system. And I think it's a little bit dangerous to then sort of translate that across much more broadly. So in terms of open questions, we have things like how is hydrogen heterolytically cleaved? What's actually the importance of all these conserved amino acids? And do we have concerted or stepwise proton-coupled electron transfer? Or do we have both? Or does it depend upon the hydrogenase, pH, and the situation, for example? To combine all this, in short, is there a universal nickel-ion hydrogenase mechanism? And really, these are the questions that my research is hoping to address. Whether I ever manage to answer them or not, I'd like to hope that we'll get some answers, but we'll see. So because sort of hydrogen activation and production involves the use of or supply of electrons, then we can use electrochemistry to control the reaction, but also to control the redox state of the hydrogenase. So if we imagine that we're not just driving hydrogenase around this catalytic cycle, we're stepping between all the different catalytically relevant states. We recently showed that you can do this in single crystals of hydrogenase. So this is work that I did during my postdoc with Kylie Vincent. So we can take single crystals of, for example, hydrogenase 1 from E. coli at any given pH, and then we can apply a range of potentials, see how the active site bands change, and ultimately, as we've shown on the sort of like the right hand side at the bottom here, come up with a potentiometric or a direct electrochemically controlled redox titration of the active sites within that particular crystal. And the important point to make from this is kind of exactly like I was saying about the catalytic cycle. There are certain states that you can make by judicious choice of potential, hydrogenase, and pH. There are certain states that you can make in essentially 100% or close to 100% yield, which is really useful for future studies. So this is fine if you want to do things in crystals, and there are lots of good reasons for doing things in crystals, not least because the spectroscopists among you will have looked at the absorbance that we're getting in the top left of that figure with all the spectra into the left of the slide, and it's really rather large. So 50, 100 millioDs worth of absorbance. It makes the spectroscopic part of the experiment really rather easy. However, there's a good reason why we want to either link the crystallographic, if you like, the structural, the in cristallo behavior and dynamics of metalloenzymes in general, but in this case nickel-ion hydrogenases specifically, with how they actually behave in solution. So that means that, okay, it's great that we can do all these in cristallo measurements, but actually we want to take a step backwards and try to improve some of our solution methods as well. And this is where a little bit of electrochemistry and some electrochemical cells come into it. So as a brief aside, this electrochemical cell might look a little bit weird right now, but at the end of the talk, hopefully you'll realize why it looks a little bit weird. So this would be a lot easier if I could use the pointer without crashing PowerPoint. But here we go. Hopefully you can see that there's a green sort of portion sandwiched in between two browny orange portions of this electrochemical cell. So the very lowest sort of portion on the slide here is really just a cap. It seals everything off. The green portion in the middle is where we stick a working electrode. We stick a carbon felt working electrode essentially to fill a large flow channel. I say large, it's about 30 microliters, something like that. And that's our working electrode. There's a working electrode connection and a reference electrode that comes through the back of the cell that you can't quite see here. Then through this sort of cylindrical part in the top sort of browny orange section, we stick a platinum wire, and that's our counter electrode compartment. So effectively what we're doing is we're making a bulk electrolysis cell here. How you make a bulk electrolysis cell work is you need a divider between the two cells. And so we use a piece of Nafion film to seal between these two compartments. And then we can flow in and out through our working electrode, so we can generate either well-defined potentials of our protein in situ within that working electrode, or we can generate a sample at a well-defined potential once it's passed through that working electrode. So the volume of that working electrode compartment, like I say, is on the order of 30, 40 microliters, something like that. So this is a massive electrode. So it had to be pretty convinced that our sort of Nafion plus counter electrode solution is actually going to work. We can test this using some cyclic voltammetry, like we're showing on the right-hand side of the slide here. This is just the ferrocyanide redox couple. It's 10 millimolar because we wanted to test the electrochemical response of the cell to destruction, essentially. Apologies that I haven't converted this to She, but I can assure you that the potential comes out about right for the ferrocyanide couple once we've corrected for our reference electrode. But the real point that I want to make here is, if you look on the left-hand side, the current scale on here is absolutely huge. So we're supporting voltammetry with sort of with current ranges up to sort of milliamps plus, using this electrode architecture.
Philip, sorry, could you, the time is running out already. Could you finish in the next 3 minutes or something?
Yeah, no problem. No problem. So the other part that we have here is, so okay, this is great. We have some cyclic voltammetry. This works if we really want to make our samples in situ inside that working electrode. If we want to do anything else with it, we need to be able to rapidly change the potential of our sample very repeatedly for a very long time. So what we've got here now on the left, we've got a chronoamperometer experiment where basically we shove small aliquots of sort of 10 microliters of solution through this working electrode repeatedly. And the initial current spike varies a little bit, but that's just because of the registration between when we pump and when the potentiostat records a value. But what we find here is that we're getting sort of 80 to 85% conversion of this huge volume within about 60 seconds, something like that. So actually we can do some quite rapid experiments. Okay, but how do we use this? Well, using this sort of pulsed through mode, we can use electrochemistry to selectively enrich a redox state in a nickel-ion hydrogenase. For example, use the nickel C state as an example here. So by slowly pulsing our sample through that working electrode, out the other end, we can pop something which is largely enriched in nickel C. We can then do the classic illumination of that with femtosecond to nanosecond pulse of 355 nanometer-ish light that triggers photolytic dissociation of that bound hydride in nickel C that you can see at the bottom, and then we can see what happens as that proton moves away from the active site. And what we've done really is we've done the same experiment, a very similar experiment to what a lot of other people have done. We just happen to have now done it on an oxygen tolerant nickel-ion hydrogenase, and we've done it in such a way where we're not really looking at a re-equilibration of potential, but we're directly just looking at that hydride photolysis and proton movement in real time. What we actually see is we see an immediate nickel C photo bleach, so far so dull, but what we see to accompany that photo bleach is an instantaneous increase in what I would call the nickel L1 state, which then decays into another nickel L state, which I would call nickel L2. So in the interest of time, what does then that really mean? So my interpretation of the various different nickel L states that are observed is kind of shown a little bit on this slide. So we start off with our nickel C. We photolyze that bound hydride and we get a transient protonation of a terminal cysteine sulfur. And then, at least in the case of the oxygen tolerant hydrogenases on a nanosecond to microsecond timescale, we're getting deprotonation or proton movement of that proton away from the active site completely, ahead of electron transfer to form the nickel Si state. So we're seeing very, very clearly a stepwise proton coupled electron transfer reaction, where for more or less there's not really any cases that I'm aware of off the top of my head in the literature. Somebody's bound to tell me that I'm wrong now, where you've directly been able to see this formation of one nickel L state followed by formation of a second nickel L state without doing some sort of fancy modeling. So I think we're starting to piece together how proton and electron movement are quite intricately related within the nickel ion hydrogenase catalytic cycle. And I guess then, in the interest of time, that's probably where I will leave things. Apologies about the minor technical hiccups at the beginning, and thank you very much for the invitation again.